So, as Johan said, I am a mathematician who for 25 years did basically only pure mathematics, and for the past 5 years or so has been involved in various applications of my area of mathematics, which is topology, outside of in engineering and in other areas of science. I'm quite familiar with some of the problems that Jan sketched out. Today, I'm going to talk about the relationship between my area of mathematical research, which is topology, and its surprising relationships with machine learning. Now, I have no background in machine learning. I've learned some things about machine learning when trying to apply topology and come up with interesting classifications of this and that in various areas of science and engineering. And so what I'm going to explain to you is something about what kind of tools we deploy. It's more like how we go through this complicated pre-processing of the data before it's consumed by the machine learning, and how you can use topology as a way to transition from complex data sets to something that is more easily consumable and understandable by machine learning techniques. So one reason for the image here is that a lot of the applications that I've done of topology have been to the area of neuroscience in collaboration with the Blue Brain project, which is an example of big science, in fact, where you have 120 scientists and engineers working together to develop some kind of a model for the brain. And that's led to greater interaction and appreciation for the role of software engineers, of the coders, of people across a wide spectrum of areas. You realize how important it is for people to develop collaboration, cooperation, and a common language across all these different areas. So, what is topology? So topology is a kind of mathematics that's not really studied in schools. Maybe most of you people don't even study it in university. Even scientists and engineers don't usually study topology. I hope that will change, because I think that topology has an increasingly important role to play in a wide range of fields. Topology is the mathematics of shape. It's the mathematics that describes the connectivity of networks. And we see networks all over the place in all kinds of areas. It's the mathematics of emergence of global structure from local constraints. So that as well is a very useful kind of tool to have available for analyzing all sorts of data in all kinds of situations. So the point is that topology has recently proved itself to be very useful for analyzing data. And so you can say, if I have a big data problem, and I'd like to try to understand something about, let the data speak to me and see what kind of shape is in the data, then maybe the person I need to call on is a topologist. Topologist who's familiar with the area of topological data analysis. So what I'm going to do today is tell you a bit in general about what topological data analysis is, what its pipeline looks like, how it interacts with machine learning to a certain extent. And then I'm going to give you two concrete examples of how my team and I have deployed these sort of topological tools for solving problems, in this case, two problems related to neuroscience. And if I have some time at the end, I'll also talk about maybe a problem related to material science as well. So there are applications across a very wide variety of fields. So a little overview of these different analytical tools that are coming from algebraic topology. So as I said, it's really all about, you have some sort of large data set. Maybe it's a bunch of gene expression data. Maybe it's a bunch of crystal structures for different kinds of materials. Maybe it's counting different kinds of trees in the forests in Finland. Somebody in the audience knows a lot about that. Then what you're going to do is just look at these large data sets, and you want to try to come up with some sort of immense dimensionality reduction to be able to say something about the shape of your data set, and hoping that the shape of your data set, which you encode in some sort of way as a mathematical signature, that you can then feed to some sort of machine learning to consume it and solve some sort of classification problem or clustering problems, things like this, that maybe this is a good way to go about it. If you don't really know, you have a large data set and you don't really know what it's trying to tell you, where you should be looking for information, let's just say, what can we say about its shape? Where do we see interesting clusters? Where do we see interesting structure? So we can briefly mention a few different methods from topological data analysis. One of the best known is probably MAPPR, which was developed primarily by Gunnar Carlson and his associates in the beginning of the 2000s. He now has a very successful startup, maybe if it's beyond the stage of being a startup now, called IAST, that uses this sort of technology for analyzing data in a topological manner. It's applicable to all kinds of data, very often to clinical data from medical experiments, metabolomics, genomics, but also to financial data. There are credit companies, there are banks that are using this particular technology for detection of fraud, for example. There's a version of Mapper that was developed by one of my PhD students. It's particularly good for analyzing gene expression data, single-cell transcriptomics. This sort of data that comes up is very popular in the life sciences now. Persistent homology, on which I'm going to spend more time today, which is probably the core method of topological data analysis, and then the recent developments along lines of graph signal processing and sort of higher dimensional versions of graph signal processing when you have some kind of a network with information on some of the nodes, and you'd like to infer information on the other nodes, and higher dimensional versions of that. But as I said, today I'm going to focus primarily on this method called persistent homology and provide you 2 illustrations of how this can be applied. So let's go into more detail on what this topological data analysis is. Now, if you had said to a topologist back in the 1990s that algebraic topology would be useful for any kind of statistical analysis, they would have said, seriously? But actually, back in 1990, there was an Italian mathematician named Frosini who actually started thinking about, maybe we can actually use topology to understand the geometry of large data sets. And in 1999, an Australian mathematician, Vanessa Robbins, who also started looking in this direction, particularly in relation to materials. It was really in the early 2000s that things got started in a serious way with Herbert Adelsbrunner and his collaborators, who started developing the technology. They were really computer scientists coming about things in a very analytical way, and then the topologists really got involved and started understanding what was the actual mathematical structure behind this story around 2005. And this is an area that has just exploded in the last 10 years or so. The number of applications, the number of articles appearing both for developing new theory and new applications has just exploded in the last 10 years or so. Okay, so what's the usual workflow? We're going to start with some sort of data set, and I'll give some illustrations later. And then from this data set, we're going to construct some kind of point cloud. So what I mean by a point cloud, I mean some collection, relatively large collection usually, of points in some sort of metric space, so say Euclidean space. So we're looking at this as a bunch of vectors, and the important thing is that we have a notion of distance between these vectors. And so we have some collection of points, we have some notion of distance between these points, and we're going to use that information to then construct what's called a filtered simple complex. Basically, it's just a sequence of different complexes, different structures, one included in the next one, and we look at how this structure evolves. And so we have maybe a little piece that looks like this, that's included in a piece that looks like this, that's included in a piece like this, that's included in a piece like this. And what we look at is sort of the evolution of this structure. So we have this passage from data to a point cloud, which I'll explain ways to do this. Get from the point cloud to here. I'll also explain more detail how to do this. And then once you do that, you sort of magically get some kind of barcode. And it really does look, what we get in the end looks something like an actual barcode that you might see on a label in the grocery store or something like that. But, and then this is going to be our mathematical signature of this data here. And the point is that this barcode is a simple enough structure that it's something that is relatively easily consumable by different kinds of machine learning. So here's an example of how you can go from data to a point cloud. So this sort of data would correspond, for example, to some kind of dynamical system where you have some value that's changing through time, and you'd like to be able to say something about the behavior of this particular function through time. So there are lots of ways you can try to analyze something like that, but the way we're going to do it is say, well, let's look at a sliding window representation of this. So what you do is you choose a certain distance along the x-axis of whatever values you're considering, and you look at the corresponding y values, and this gives you, and you do that for a fixed distance between the points on the x-axis. You choose a certain number of points you're going to consider on the y-axis, the corresponding function values, say 3 of them, and those are going to be 3 coordinates of a point in 3 space. And you do that, you choose you start with a different value of x, and you choose 3 values here. You look at the corresponding function values, and you map those to a 3-dimensional space. And you do that for a whole bunch of choices of points along here, and it gives you a whole bunch of points in 3 space. This is called a time delay embedding of this curve into some sort of 3-dimensional space. The point is that it takes this curve and produces a sort of a point cloud. It gives us a collection of points in 3 space, which are encoding a lot of the structure of this curve here. And what we're going to do is apply, now we have something to which we can apply the tools of topology once we do this one more step, which is to associate to this point cloud sort of a sequence of complexes. So now, if you know something about topology, you say, well, this is just some sort of discrete set of points. How am I going to be able to say anything about the structure here? This is not very interesting. The point is that topology, even though it usually is studying sort of continuous structure of things, and this is like some sort of discrete object, what can we say about that? The thing is that by varying the scale at which we're looking at the relationship between these points, we can actually start to see structure. We can use topology to start to detect the geometry of this object. And you say, well, this looks like it should be information that's lying on kind of a circle, so maybe we should be able to say something circle-ish about this data here. Why are we seeing this? This circle here is really picking up the fact that here we have this very periodic kind of behavior that's represented by the fact that we have this circle shape when we've done this delay embedding of the data. Okay, well, once we've gotten to a point cloud, then what do we do? Okay. So what we're going to do now, so this is my collection of points, and what we're going to do is start growing little balls of a fixed radius outside of these points. And when 2 balls intersect, we're going to connect the points with a little segment. So we're looking at balls of radius 0.3, then we have not too many balls intersecting, so not too many of them that are actually connected by lines like this. But we're going to increase the radius little by little, and as we increase it, there are more and more edges that are connected, and it starts forming shapes. We start seeing these circles, we start seeing more and more things that are getting connected together. And we see how this evolves as we increase the radius gradually. 0.5, 0.7. Now we start to see, we're also going to start picking up the fact that there's kind of a circular structure here. There was a little circle here, there's sort of a bigger circle here, that now, at the next stage, we've increased the radius to 0.9. This little circle is disappearing, but now we're seeing that this other circular behavior is here. So by looking, not thresholding, but actually looking at a whole sequence of different distances, this allows us to detect structure at different scales in this data set. If we increase even further, then the little circle over there has now disappeared. Now we have edges between all of these guys. And even further, and even further. And in the end, we have edges between all of the points because all of the balls are intersecting now. And so what we do is we take our point cloud and we look at this sort of sequence of collections that we get by connecting points with edges when the balls around these points actually intersect for the different radii. So that's the way we go from the point cloud to these sort of nested complexes. Now we need to get to a barcode. So the barcode is going to look something like an actual barcode that we see in the grocery store. So here we have our collection of points. There are just 4 of them. There's a toy example for illustration. And what we're doing is slowly increasing, I don't have the actual balls that are being drawn here, but you can think about the growing balls in each case that are starting to intersect and leading us to add in lines or to fill in triangles when we have all 3 edges that are already there. So when we do that, what happens? So in this case, at the first stage, we see that we have 4 points, 2 of them are connected. So we have 3 distinct pieces, 3 what we call connected components. So we call that Betti 0 of this complex. It's the 0th Betti number. It's counting the number of connected components. So here we have 1, 2, 3 connected components. And so it gives us 3. And we don't have any cycles in this story. So that would be just say in that case that Betti 1 is case. So Betti 1 of a complex is counting how many cycles, how many loops you have in the complex. In this stage, we now again have, if we look at the number of different components or different disconnected pieces, we have 1, 2, 3, 4 disconnected pieces. So we count that here. And now we have a loop that has appeared. So we say we're counting that loop right there. At the next stage, we now have only two different components, because we have all these things that are connected together, this one that's still disconnected from the others. And then we have now two loops as well. Here we have, this one has been filled in, so it doesn't count as a loop anymore. And here we have two, so that's two. Here we again have two different disconnected components. We have one piece here and one piece there. And then we have now just one loop. So what we do as we go through this complex is we're keeping track of the number of disconnected pieces, these disconnected components, and we're keeping track of how many of these loops, how many of these cycles there are in there as well. There are higher dimensional versions of this, where you can think about how many, do I have an empty tetrahedron, do I have empty sort of higher dimensional versions of this, but let's stick to something that's easily visualizable for the moment. And so the way you can encode this information instead of just writing down sequences of numbers is through these sorts of barcodes. So what does a barcode do? So here are these different steps of the process, 1, 2, 3, and 4, that we put along this axis. And then we keep track of when different the components appear and then disappear. So for example, we know that in step 1 we had 1, 2, 3 components, so those get 1, 2, 3 bars that stop here. One of the components gets joined up with the others there, but the two others continue, and you have other ones that start in the second step. And so you have these barcodes these bars, that start at certain places and end at others. We call these sort of colloquially the birth and death points of these different bars. And for dimension 1, for the cycles, so these are the values of Betti 1. We have Betti 1 at the beginning, which was just 0, so we have nothing there, but we have a bar that started in filtration 2, when we had a 1 there, and it continued just the second step. And then we had other ones that started here, one of them in the continues forever, and the other one which was then filled in at the fourth step. So we have this process of taking one of these nested complexes and converting it into one of these barcodes. And these barcodes are now these sort of topological signatures that are easier to work with. They're very discrete things, are the things that you can now actually use if you're trying to do some sort of classification or something like that. So here's just another example showing more little dots that are slowly being connected and then forming a circle and then getting all filled in. So this is just the kind of barcode we get. The solid lines here are indicating the number of connected components, the number of disjoint little pieces you have. And the dotted lines are when you have the creation of some cycle, well, like this, one of these loops that eventually gets filled in. So this is just the kind of thing that shows up when you're doing these sorts of barcode computations. Okay, so what are you going to do with a barcode now? If you've actually gone through this transition where you start with the data, you feed it into some machine that's giving you a point cloud, and from a point cloud you get this nested complex, and from the nested complex you get the barcode. So this is this whole processing pipeline of topological data analysis from persistent homology. So what do you do? Well, one thing you do is to say, well, this is kind of this sort of barcode is a bit of an odd gadget to work with. It's not the sort of thing we're used to human reading. It can often be easier and totally equivalent to express this rather than as a barcode as what's called a persistent diagram, which just records the birth and death points of each bar. So here we have a bar that starts in 0 and ends at 2, and that corresponds to just a point, 0, 2. Or we have a bar that starts in 0 and goes to 4, that corresponds to a point 0, 4. And similarly from 1 to 2, or 1 to 1.5, that's this one. And then we have ones that go from 2 to 3 and so on. So you can go back and forth between thinking about things in terms of barcodes, which is typical on the west coast of the United States, and to thinking about persistence diagrams, which is the language of the east coast of the United States. For some reason, the two coasts of the United States have different approaches to this. I think it depends on who the guru is. Carlson on the west coast, and I don't know exactly who on the east coast, but anyway, they each have their own language. These are equivalent languages. Some are easier to work with than others. For some of the approaches to machine learning that I'm going to talk about in a moment, it's easier to start with barcodes. With others, it's easier to start with persistence diagrams. But these things are strictly equivalent. And this is maybe a little bit more human readable from my perspective. But then again, I'm not from the west coast of the US. OK, so barcodes and persistent diagrams. That's the sort of output we're getting from persistent homology, which is a flavor of topological data analysis. Now, one thing that's really important is to know that this sort of process is stable. If you change your data set a bit, if you perturb it a bit, so that maybe your measurements weren't quite right, there's always going to be noise, right? Out in the real world, there's a lot of noise, particularly if you're working with biologists. And so you have to take that into account. You have to hope that whatever method you're introducing is not that sensitive to noise, not that sensitive to error in measurement. And so you want this whole process to be stable, so that if you have two different data sets which are only perturbed one slightly from the other, then the resulting barcodes or whatever output you're taking from the topological data analysis is also relatively similar. So it turns out that there are reasonable notions of distance on the set of all barcodes or on the set of all persistence diagrams, which are, for those who know what we're talking about, sort of earth mover type, Wasserstein type, in terms of how you calculate the distance between these particular signatures. And so the point is that most ways you have of building up this pipeline for topological data analysis are actually stable with respect to this choice of distance. So you could say, if I have two data sets which are close in some sort of perhaps Hausdorff distance sense, or you have some appropriate notion of when two data sets are actually close to each other, then you'd like the corresponding persistence diagrams or barcodes also be close to each other. And that is actually the case for most of the instantiations we care about. And so that means that whatever analysis you do on the output of the pipeline, when you're looking at these barcodes or whatever, will give you some sort of valid information about your starting point, which was the original data set. Okay, some practicalities. So there are, in fact, large software libraries that are useful for doing these sort of TDA computations. There are a few that are mentioned there, some other ones that I didn't put up there. There's a method for doing these computations called Irene. There's a method, there's another thing for doing these computations called DIFA. So there's a whole bunch of libraries, most of which are open source, and which are relatively more or less user-friendly, depending on whether you were looking at somebody who was a pure academic researcher or somebody who had some software engineering experience and actually cared about the user. And then more and more people are developing sort of inverse analysis tools to say, okay, I've noticed within my persistence diagram or within my barcode that here are some significant areas. Here are some significant differences in these barcodes. How can I translate this back into saying something about my original data set? So there are researchers, I think primarily of the work of Yasuhiro Oka and his collaborators, who are developing really powerful inverse analysis tools to be able to say, okay, here is what's going on with the persistence diagrams. What does that mean about my original data sets? And they've had some really great applications there to material science, for example. So there are practical tools here. But now, this is supposed to be about topological vendors and machine learning. All I was telling you so far was how to do some sort of fancy pre-processing of data, to take the data sets and convert them into these topological signatures. But what we want to do now is take these topological signatures and somehow use them to satisfy some sort of classification problem, maybe do some sort of unsupervised clustering, do various things. So how does one go about that? Well, the problem is that if we just stay in the world of persistence diagrams and barcodes, this is not a good space in which to do any statistics. You don't know how to take an average of barcodes or an average of persistence diagrams. You have to, if you really want to do some statistics in this case, you can do it there, use some sort of kernel methods having to do with using the distances in these spaces, but it's still not ideal. So what people often do is some sort of featurization. So they choose some way of taking the set of all these barcodes and persistence diagrams and associating to them some sort of vectors in a vector space that has to have an inner product, so you have a notion of distance there. So maybe a Hilbert space, or you have some way of mapping to a space where you can actually do statistics. You can actually compute averages because you're in a vector space, so you know how to add things and you know how to multiply things by scalars and such. So this is what they call featurization, and there are many approaches to this. This is like a really blooming area of research in in topological data analysis, how are we going to featurize things so that we can actually do some computations, actually do some statistics? So one approach, which is probably the easiest, is what's called Betti curves. So here I have a barcode. Suppose I'm computing, I have my complex, and I'm looking at the wholes of dimension k. So you could think of that in the case k equals 0 as just being, I'm counting the number of connected components, the numbers of disconnected pieces that I have in the complex. Or in the case of k equals 1, it would be the number of loops or cycles that you have in your structure. And so these are my different filtration steps, 0 to 5, for example. And I say, OK, I have 2 bars that start in filtration 0, one that ends at 1, one that ends at 3, another one that starts in 1 and goes to 2, another one that starts in 4 and goes to 5. And say, OK, what am I going to do? To compute the Betti curve, what I'm going to do is for each filtration, I'm just going to count how many bars there are. So in 0, there are 2 bars. In 1, there are 3. In 2, there are 2. In 3, there's 1. And in 4 and 5, there's also 1. It's a different bar, but it's still there. So just by doing this counting, of course, you're losing a lot of information about the structure of the barcode. But it turns out that it's still a relatively powerful, relatively useful thing to do. So what do I get out? So I get this curve where here on the x-axis, I have my different filtration values. And then on the y-axis, I'm just counting the number of bars that are present at that filtration value. So just get a simple curve like this. Obviously, you're losing information, but it still turns out to be something useful to study. Then, once you've reduced the problem of studying barcodes to studying curves, now in the space of curves, OK, you have all sorts of measurements you can do. You know how to add curves. You can multiply a curve by something. So now you're in a space where you can actually do some statistics. However, you would like to use some notion of distance between curves. There are various approaches to this. Here's an example of how we go from one of these nested complexes to a Betti curve, a little toy example. So here's my little complex. It just consists of 4 vertices and 5 edges that I'll suppose are different lengths. The distances are different. I say alpha less than beta less than gamma. So when I'm at threshold 0, I just have the 4 points. Everything is too far apart to be connected. When I get to alpha, now these two are connected because the balls be intersecting. When I get to beta, I'll add in these edges. And when I get to gamma, I've added this edge as well, which means I'm going to actually fill in these triangles. So I have a nested complexes like this. And so if I'm looking at the Betti 0 curve, so I start at 4, and then when I get to alpha, it drops down to 2. And then when I get to beta, it drops down to 1. And it stays at 1, because I always will have one connected component from then on. And in dimension 1, I have no cycles, so 0, 0. Then I have 1 for a little while, and then it disappears because it gets filled in. So that's the kind of way you can encode this structure of these Betti curves. Basically, we're cheating when we sort of when we do these interpolations as either step functions or as I did it with just drawing segments between the points, because all we really have are the points at certain values. In this case, actually, we know that it's constant between 0 and alpha. So in any case, we've reduced now to the point where we have a curve, and now we're in space of curves, we can actually do some statistics. Another thing you can do is dimension reduction even further. You can just say, well, if I have a curve, I can just do things to the curve, like measuring the area under the curve, or measuring the maximum value of a curve, or finding the value on the x-axis where the maximum is achieved. There are various little tiny numerical invariants that you can extract from these kind of curves that turn out to be surprisingly useful for various machine learning tasks. So this is like, we started with this very complex data set. We go through this long process, data set to point cloud, to nested complex, to barcode, to Betti curve, and then now we're extracting these very simple invariants from the Betti curve, and they turn out to be remarkably useful, as we shall see. So one other approach, which actually it turns out to be related, part of the same family of approaches to featurization as the Betti curve, is something called persistence landscapes, which in this case will start with a barcode and convert it into something that actually looks like a mountain landscape. Now this method was developed by Peter Bubenik, and Peter Bubenik was a postdoc with me in Switzerland around the beginning of the 2000s. And I always wondered whether he wasn't inspired to create the notion of persistence landscapes by having spent 2 years living in Lausanne, Switzerland, and looking across Lake Geneva at the French Alps. But in any case, it's a very useful sort of featurization. Again, you're in the world of curves. You have just you associate to a barcode some collection of curves, and then you can define a distance between the curves by using either some sort of L2 distance or some other version of distance. In any case, you're in a space of curves, and now you can, again, do statistics, and it turns out to be a very useful featurization of these barcodes. And in fact, both the Betti curves and the landscapes are special cases of something that was recently developed called persistence curves, and I realized I forgot to mention the reference here, Leon Lawson. So there's like a wide variety of variants. You can start with the barcodes and extract from it curves in many different ways by taking the birth and death values of the bars and then performing various functions on that and adding things up or taking maxes and so on. But these are all part of one big family of ways one can extract curves from these particular barcodes. Something that's a rather different approach is what's called persistence images, which is also a very useful way of studying persistence diagrams and, again, moving into a world where you can actually do statistics. So the idea is that if you start with a persistence diagram, like this one, you replace each point instead by some sort of Gaussian kernel, which allows you to smooth things out. And then you discretize it, you pixelize the result, and that allows you to associate to something like this, a matrix, which we can represent by some sort of heat diagram like this. And once you have a matrix, then you can do all kinds of statistics. You have all kinds of tools at your disposal. And this is just giving you some examples of various persistence diagrams and their corresponding persistence images. So just smooth it out, discretize, and you're in business. You can do all sorts of interesting statistics now. Then, once you've done the featurization, you're finally ready to apply machine learning methods. Machine learning methods that have already been applied so far are basically anything you would like. The ones that I know of best having been used: decision trees, random forests, support vector machines. Convolutional neural networks are starting to come into play. And I'm in discussion with a group within the Blue Brain project of people who are working on applying graph CNNs as well to this sort of data, because a lot of the data that's coming out is actually naturally of a graph form, and you'd like to exploit that by using graph CNNs when you do your machine learning. This is not an easy problem to solve. Now we're making good progress along those lines as well. There's really a wide variety of machine learning techniques that can be applied once you have featurized your data. Okay. So let's see some concrete examples of this particular pipeline. The first one I'm going to talk to you about is about using topological data analysis to characterize neuron morphologies. This is work that's been published in the last couple of years, led by a PhD student within the Blue Brain project named Lida Canari, in which I've been happy to collaborate. So here, this picture is meant to illustrate a little bit of the diversity of the shape of neurons. So these are actually what are called pyramidal cells. So they're the biggest excitatory cells in the cortex of a rat. So if you think about the cortex of a mammal, it's actually constructed in 6 different layers, from the surface of the brain to the inner part of the brain, from here in layer 2 to layer 6. So we see there's a wide variety of shapes and sizes and branching structures and so on. The structure, the shape of a neuron is very important for its function. So really has a strong influence on how it functions, what its role is in the brain, and also in how it connects with other neurons. So understanding how to classify neurons in terms of their shape is an important problem in neuroscience. Okay. So what we wanted to do was to find some sort of objective way of going about this classification. Now, of course, neuroscientists have been trying for decades to find good ways of classifying neurons. There are people who are known to be expert classifiers and who provide subjective, I'm looking at a neuron under the microscope kind of classifications. The problem is that there have been experiments done where you give the same set of reconstructions of neurons to different experts, and they give you different classifications. You can take the same set of neurons, give it to the same expert on 2 different days, and get different classifications. So this is not ideal. You'd like to have some method that didn't change its mind from one day to the next. Now, as we'll see later, the experts actually do a pretty good job, even a very good job, but still, there are some ways that you'd like this to be a more objective process, particularly since you want to be processing a lot of data. Now this gets to one of the things that Jan was talking about in his talk, where he said a lot of the effort goes into data collection. I need to explain something a little bit to you about where the data is coming from. So if you want to know something about neurons from a brain, what do you have to do? You have to look at a bit of brain. So you take a rat, you sacrifice the poor buddy, and you take a slice of his brain and you look at it under a microscope. And then some poor PhD student or postdoc has to spend hours and hours and hours painstakingly recording the 3D coordinates of a bunch of points along this neuron, as well as telling you which particular pairs of points are contiguous, so that they can record the structure of this neuron digitally. So there's a huge amount of effort that goes into data collection here, and it turns out to be, it's relatively easy to do with these big pyramidal neurons. It can be very hard to do when you have smaller neurons. So it's really data collection here, data processing, data cleaning is an enormous problem. But in any case, once we have some digital reconstructions, we'd like to go about doing some of this classification. So what are we going to do? We're going to find a way of associating to each neuron morphology, and basically this method will apply to any kind of branching structure, a persistence diagram. Now we know we can't do statistics with persistence diagrams, so we're going to find a way. We're going to use persistence images, in fact, to featurize these persistence diagrams and then do classification methods. Now what we're looking in particular in these neurons is we're looking at what's called the apical dendrite. It's sort of the biggest part of it. So the neuron has 3 major parts. It has a dendrite, which is for incoming information. It has a soma, which is sort of information processing, is the cell body. And then you have the axon, which goes out. Axons are very, very hard to reconstruct in the lab. So we're focusing here just on the dendrites, in particular the apical dendrites, which are the dendrites which usually extend towards the surface of the brain. So we're going to do this, apply these methods. We're going to find some way of associating to any tree-like structure, and in particular to apical dendrites, persistence diagram. So the idea is the following. We're going to take our neuron, and we're going to use as a reference point its soma, so the cell body of the neuron. And we're going to look at it as a tree with whose root is at this soma. And we're going to use basically persistent homology, looking at just components. We're going to break this thing up into branches, taking the branching structure, so the combinatorial part of the branching structure into account, but also how it's embedded in space. As a way of translating this tree structure into either a barcode or a persistence diagram. We're going to prefer persistence diagrams here usually because we're going to look at persistence images. And so what you do is you sort of break it up bit by bit and break it up, and these bars here are corresponding to the different branches of the tree, and which are sort of broken off as you move from the outside towards the root. And at the very end, the big bar that's left is sort of the one that's the very longest, from the very farthest reach all the way to the root. And then we translate this barcode into a persistence diagram and study these persistence diagrams. So in more detail, if we have a very stylized neuron, whose root here is here at R, I'll explain a couple of steps of the process. How do we break this up into branches? So we'd start farthest out from the root, at H here, and we go to where it reaches its first bifurcation point. And because H was the furthest out, we say, well, the other branch that was at the bifurcation point, we're going to break that off and turn it into a bar in our barcode. And then we continue along our way till we get the next bifurcation point, and say, okay, well, here was another guy, E to D, but E was not as far out as H, so we're going to break off that branch, E to D, and we'll take that as a bar. We keep going to I, and here we have another bifurcation point. Now we have to backtrack quite a bit. Here we say, okay, at B, we had another bifurcation point. A was not as far out as C, so we break that one off. That gives us A to B. And then if we compare the path from C to I with the path from H to I, H was further out than C, so we break off this branch C to I. It stays there. And in the end, all that's left is this very long path, or the longest path, from H to R. And so that gives us this this barcode here, that is then translated into a persistence diagram. In this case, the points are under the diagonal because we're taking our birth points to be the points farthest out from the root, and where they break off is usually closer, except in the case of A, because sometimes if you look at a neuron, the end of the branch is actually closer to the soma, to the cell body, than its bifurcation point was, and then you get a point above the diagonal. So here we can get points above and below the diagonal in my persistence diagram. So anyway, we have this process for associating to any branching structure like this, doesn't have to be a neuron, a barcode or a persistence diagram. We validated this on random trees. So we had a way of generating random trees where we could fix the tree depth, the branch angle, branch length, how random the branches themselves were. And, oops, that was too fast. There we go. And then we did a comparison where we were looking at a dendrogram, and what we're looking at in this heat map is the distance between the persistence diagrams, which is one of this bottleneck distance, is one of these earth mover type distances that we were looking at, and we saw that this actually does a pretty good job of just using this distance to do this classification. I think that the accuracy was on the order of 90% for doing this classification. So we said, okay, it's looking good on random trees. Now let's try a toy example. A toy biological example. So this toy biological example is to say, okay, I'm going to look at neurons from 5 different species. And obviously, if we look at these neurons, just as humans, looking at them with our eyes, we can say, even if we're not neuroscientists, we can see that these 5 are different. And if I give you an arbitrary new member of each class, you could probably figure out what class they went into. Wouldn't be a problem. But the point is to see whether the machine can do as well as we can. So we took a population of, a collection of neurons of these different types, looked at the associated barcodes, looked at the associated persistence diagrams and persistence images, and in this column we have the average over the entire collection. And they say, okay, can I actually train some sort of machine learning method to classify these neurons based on their persistence images? And it turns out that works very well. So here we were just using a decision tree, actually, to do the classification, and ended up with quite high accuracy. So this is the confusion matrix. And we see that, there was some confusion between mice and fruit flies, of all things. But other than that, it was doing a very good job of the classification. And the training set didn't even have to be all that big before you reached a relatively decent level of accuracy. And more recently, somebody went back and looked at, well, when you're using the decision tree and you're applying a decision tree to the persistence image, how many points of the persistence image are you actually using? And it turns out that of those, I think it's on the order of, 10,000 pixels or something, you use 18. Actually, only 18 of the pixels allow you to compute this difference. In any case, the classification works very well. But that was kind of a cheat because you could do it by eye really easily. So instead, what about a harder problem? We are actually looking at all the pyramidal cells in the brain of the rat. So that was the image I showed at the very beginning. So here we have the cells arranged by layers. Remember I told you there are layers in the cortex. And so you have some of them, they're concentrated in layer 2. So you really look at it, in what layer is the soma or the cell body found? And so these are schematics of how the subjective classification was done by the experts. So the ones who just sort of eyeball it. So there are other people who don't feel as confident in their experts, in their ability to eyeball things, and so they look at what are called morphometrics, which are various numerics associated with these neurons, and they try to classify them using PCA or whatever. It never really works very well. So let's try our objective classification. So we applied this pipeline of associating with our topological morphology descriptor, so these persistence diagrams to these guys, and then applied machine learning methods. When we did that, we found out that there were a few mistakes that the experts were making. For example, in layer 3, that there was some confusion between 2 different types, that by applying our objective methods, we were able to sort out very well and come up with sort of more coherent structural picture of what was going on. And now if I have only one type that was really ambiguous, ending up with very good classification. In layer 5, sorry, this is probably hard to see, there were considered to be 4 types of pyramidal neurons, but with our persistence images there, we were not seeing any kind of distinction. And there turned out to be a lot of confusion between these 2 types here when you applied any kind of supervised methods. So in the end, it turns out that what's going on is that in layer 5, there really weren't 2 distinct types, but there's really a continuum of types. If you look at the extremities, of course, they're quite different, but there's sort of a convergence in these different types of neurons. So this enabled us to make clear that there was more structure, that the situation was more complex than what the experts had thought. Okay, so briefly, I want to talk about a quite different, but still related to neuroscience, application of these methods. So using topology to explore dynamics. So with the previous case was something that was very geometric. We're looking at trees. Now we're going to see how we can change a problem in dynamics to something that we can study by these methods. So I mentioned at the beginning something about using delay embeddings of curves as point clouds. This approach is going to be quite different, where we use surprisingly simple methods, actually, to come up with a very powerful classification with high accuracy. So when you're thinking about the dynamics in a network of neurons, it's very complex. It's very difficult to quantify what's going on when neurons are firing and there's activity moving through a network and so on. And what you'd like to do is have some way of coming up with some kind of quantification or classification of this kind of activity. So why would one care about something like this? Well, there are many reasons, of course. I mean, neuron dynamics has to do with information processing, and we want to know more about how the brain processes information. It's a huge mystery. Also, different patterns of activity can correspond to different roles of the brain. So the hippocampus is responsible for both memory and navigation, and these 2 things are really related. And there are different patterns of activity that correspond to these different roles of the hippocampus. Also, if you're curious about different types of neurological disorders, these are often characterized by abnormalities in neuronal activity. Particularly, everybody knows the case of epilepsy, that you have like a sort of electrical storm in the brain, but also in Alzheimer's. There are changes in the electrical activity in the brain that you'd like to be able to quantify and maybe predict. So the goals of our project were to try to find some sort of automated classification of neuronal dynamics using these methods of topological data analysis. And the construction that we do depends on determining some kind of measure of similarity in the spiking behavior of neurons. So neurons, when their membrane potential hits an appropriate level, they will emit what's called an action potential. So it's like a chemical electrical signal that they emit, and this is the way information is transmitted around the brain. And so what one records in a neuron is when the neuron emits what we call a spike, like that, and you have what's called a spike train, where you look at the sequence, it's a time series of spiking behavior of the neuron. And so you'd like to be able, you have various ways of describing similarity between spike trains of neurons, and you can vary this measure and take these different possibilities into account in topology. So what we did was construct 3 artificial networks of a certain kind of model neuron, which is called a leaky integrate and fire neuron, where you can change the connectivity, you can change the delay in the structure, which is, you can think of as a delay in the network, and the strength, the transmission strength. You're going to simulate the activity over a range of different parameter values, so we can control different parameters in our networks. And then from the simulated activity, we can build different weighted graphs. So these are networks with different weights along the edges. And then we can apply persistent homology to that, because when you have a weighted graph, you can think of it as a point cloud, where the distance between 2 points is given by the weight, or 1 minus the weight, along the edge. And then you can extract simple features from these invariants, as I described before, and it turns out that we can use those to train a classifier to predict dynamics. So we did a number of experiments. In fact, we did 280 different simulations for each network, where we varied the parameters we could play with, one of which was a sort of conductance, and another which was a sort of rate of excitement of the network. And so we had a range of 28 parameters there, so we're taking integer values, and we did 10 trials of each one. And then we recorded the spiking time, so these spike trains. When is a neuron emitting an action potential? And we recorded that over time for each of the neurons, and also looked at when all the different neurons were firing together. And ended with 4 different, very distinct dynamical regimes. So you could either have, in this picture here, we see along this axis you have all the different neurons, and there's a little tick mark every time it fires. So we see this very regular activity in this. Here, this is a histogram counting how many neurons are firing at each time. So there we have something that's very synchronous. There's also something that was synchronous but irregular. A lot of the time nobody's firing, but when they do fire, they all fire together. You have something that's it's not coordinated at all. It's very synchronous, very irregular. And the last one was periods of non-activity and then big bursts, and so it's very unpredictable. We just call this the alternative regime. And so the 3 different networks we set up had different patterns depending on these parameter values we could choose. So we had 3 different networks where the different kinds of regimes you had depending on the conductance and this sort of rate forcing we had. We ended up with pictures like this, where you would see the different regimes according to these different values. And what we want to do is be able to predict the regimes for these different things. So how do we do that? Well, we have these spike trains associated to each neuron, and so to measure a distance between 2 neurons, we use different notions of similarity between these spike trains, which we could use either person correlation, or there were various other examples in the literature of ways you could measure similarities between these spike trains, which is both spike synchronicity and spike distance. Once you have these weighted graphs, as I said, that gives you a notion of distance between points. So this is an example of how these different measures of similarity look quite different. So they're not measuring the same thing. So we associate the point cloud of vertices with distance given by the weighted path distance. We can look at the associated nested complexes, the Betti 0 and Betti 1 curves. We record the area under the curve, the filtration value, which it starts to decrease. So we're extracting these very, very simple numerical invariants. For the Betti 1 curve, the area under the curve, and the global maximum. So we get 12 features extracted for each of the simulations. And from those 12 features, we actually end up doing very good classification. So let's see. I don't have much time, so I will just skip through the details of the classifier. It's a support vector machine, which we trained in an appropriate manner. One of the reviewers in the paper said, couldn't you just use this linear classifier? He said, oh, maybe. But anyway, we used a support vector machine. And when we did that, there we go. Then we assessed the accuracy, appropriate sort of cross-validation. In the end, we actually had fantastic accuracy for the classification using these very simple topological features. And so we could even use one version of the network to train and test on another version of the network and still get very high accuracy. So this is something quite universal that we were detecting with these very simple topological features. Also, we could use the distances were complementary. It was a good idea to use a combination of them, and it led to better performance. So we were able to practically perfectly predict network regimes from a few features extracted from persistent homology. It could even predict with high accuracy the dynamics of other networks than that on which it was trained. And that it was good to use different notions of distance to try to come to an appropriate conclusion. And with that, I would like to thank you for your attention and your patience with mathematics on early in the morning. Tack så mycket.
Thank you. Comments or questions?
That was a highly useful presentation. So you mentioned the connection to Wasserstein distances and the optimal transport problem. Can you elaborate a bit on that? Exactly how that, and do you actually use, over recent years, people like Marco Couturier and others have developed really fast methods for actually solving these problems?
Right, so I mean, since we do a lot of these computations of these optimal transport type distances, I mean the Wasserstein distances and so on, I'm in the lucky position of being able to tell my postdocs just to do things. I don't actually have to. And I know they're constantly looking for faster and faster algorithms to do these sorts of computations. This is really important for this area. And to understand well also as much as we can about the theory of these kinds of distances, to be able to understand better what it is we're actually computing when we look at a Wasserstein p distance between two persistence diagrams and so on. So I think there's a lot of overlap, and it's interesting because I don't think this is an area in which algebraic topologists would otherwise have been very interested, thinking about optimal transport. So it's really cool, I think, that this is leading to more interaction with between different areas of mathematics, which one would not have expected at all 10 or 15 years ago.
Okay, just a question. I mean, we're looking at, in many cases, very rapidly changing data with features appearing. In the brain data, you have elements of that, but also when the underlying topology changes. Could you elaborate a little bit on the pros and cons of using barcodes for that representation or persistent graphs? What would you find more intuitive?
So another problem that we're studying is the problem of synaptic plasticity in networks of neurons. So there you have a weighted graph that is changing with time. The weights are changing in time, which is also one we might get in this dynamic situation. And in that case, what we're doing to start with is computing a persistence diagram, sort of binning time, and then doing a persistence diagram in each time bin. And it actually creates a persistence movie, because you see how the diagram is changing with time. And you can sort of put your finger on it and say, look, there's an interesting area that's developing. And then you can do something like using these new inverse analysis tools of Hiraoka and people like that to be able to say, okay, to what part of the data is that evolving interesting feature corresponding? So, yeah, I mean, it ends up being more of a multidimensional thing. So there's another approach to persistence called multidimensional persistence, which is a much harder problem than just looking at filtrations along one dimension. And then one has to, there are various tools that have been developed for tackling these sort of multidimensional persistence problems. And one could also, in this sort of time-varying situation, approach this not sort of time bin by time bin, but do it in a more integral way and use multi-persistence in that case.
So you would see time as just another dimension in that
You could. That would be another approach to doing that.
Right.
But it turns out that, I mean, it's much easier to do the time slices, and even that is already giving us interesting information. If then, it would be interesting perhaps later, at a later stage, to go back and say, okay, let's try to do this from more of a multi-persistence point of view. Once we've extracted everything we can from doing it the easy way. I've really developed some tendencies to be an engineer over the last few years. An appreciation for the engineering approach.
So I have one question. So can you tell in advance for a particular data set that topological methods might be useful compared to more traditional deep learning, say?
I think that's really, so far a lot of that is gathering experience and starting to see. I mean, I have come across some data sets where it turned out that were not very useful at all. It's really, really hard, for example, with MRI data to use persistence techniques. There was an article that was on bioRxiv just the other day talking about what you can and can't do with persistence methods when you're looking at fMRI data. Part of it has to do with how noisy it is, part of it has to do with the kind of data it is, and there's a whole question, do you take into account negative correlations? If so, how? And so there are some problems that are still kind of resistant. I don't know if they can't be attacked, but there's some that are much easier than others, that's for sure.
One last.
Last, yeah.
One last question. Need coffee also.
I'm available for questions during coffee break too.
Very interesting. I was wondering in the connection of what we looked at yesterday from you with the explainability of the neural networks during training. Have you thought about applying these methods to look at the artificial neural networks, not only the actual neural networks?
So absolutely, and this is a question I get a lot when I talk about this. So I think that so the two things I was talking about today with neuromorphologies and dynamics in artificial networks of neurons, I mean, particularly the second one might be useful for thinking about artificial neural networks. But there are other techniques that we've developed for when looking at the blue brain model for the brain, where we've applied another collection of topological techniques that I think would definitely be useful and interesting to apply to, studying CNNs or studying other kinds of artificial neural networks, particularly recurrent neural networks of various kinds. And I would love it if somebody who in that community picked up these tools and said, okay, we know what this gives us when we look at this digital reconstruction of part of the brain. What does it look like when we do a comparative study for these artificial neural networks? I think that could be really interesting, and there's a lot to learn from that.
Thanks again.